Hello all, welcome back to the BioRock channel. In this video today, we'll be dealing with the Mendelian disorders and the one of the Mendelian disorders which comes under the category of autosomal recessive disorder, that is going to be the thalassemia. And before I jump into this topic, children, I would like to clear that there are two different types of hemoglobinopathy, which is nothing but the disorders where there is either quantitative or qualitative reduction in the production of the globin chains in the hemoglobin. Thalassemia comes under the category of a quantitative abnormality where the quantity of the globin chains in the hemoglobin is going to get reduced. Okay, so this thalassemia is going to be a Mendelian disorder which comes under the autosomal recessive disorder, and it is a quantitative abnormality in the production of the globin chains. Usually, if you're going to look at the red blood cell, the erythrocytes, we find that they are going to comprise hemoglobin, right? So if you look at the hemoglobin, there are two alpha chains, okay? So you can see this as one alpha chain, the other alpha chain, and it is being conjugated with the other two beta chains, okay? So there are two alpha chains and two beta chains. So it's a conjugated protein chains, which are being called as globin, right? Hemoglobin, where globin is going to stand for the protein and heme stands for the iron. So where is the heme group is, this is going to be the heme group. You can see that each globin chain is having its own respective heme group, right? So one, two, three, four of these heme groups, where you can see the iron, the red one that you can see here is going to be the iron, and this structure is going to be the heme group. So each and every globin chain, the two alpha chains and the two beta chains respectively, have their own heme groups as well as the iron. Okay, this is a general hemoglobin that is being found in normal humans. But actually, if you look at the hemoglobin in the RBC children, in the fetal stage, you do not have the alpha chains, but instead of the instead of the beta chains, sorry, instead of the beta chains, in the fetal stage, we have the gamma chains. Okay, and in the adults, we can see this sort of hemoglobin that you're seeing on your screen with two alpha chains and two beta chains. And along with this, we have a minor portion of another type of hemoglobin also in the adults, which is regarded as a hemoglobin A2 type. Okay, this is hemoglobin A type. We have hemoglobin A2 type also, where you have instead of the beta chains, we have the two delta chains which are being present. Okay, I'll give you the details about that. But I wanted to give you an idea about the hemoglobin present in your red blood cells. So just please do remember about these globin chains, the protein. So as thalassemia is going to be quantitative abnormality where the globin chain production in quantity is going to be reduced in this autosomal recessive disorder. Okay, so this is a Mendelian disorder that we are learning in our syllabus. So now what I'll do is I'll go into the details of this. For that, let me share my board with you all such that I can give you the details of the thalassemia. Okay, so I'll give you the details cut to cut, which will be important for our NEET examination. So I'm speaking about the Mendelian disorders. Mendelian disorders. Amongst that, today we are learning about the thalassemia. Okay, we are learning about the thalassemia. So, children, before I enter into the disease thalassemia, let me first give you the picture of hemoglobin. What are the types of hemoglobins that are being found? Okay, types of hemoglobin. So, generally, we find the hemoglobins as hemoglobin F type. Next, we have hemoglobin A, which is generally seen in the adults. Next, we have is hemoglobin A2, which is a minor fraction of hemoglobin found in adults again. In the hemoglobin F, F stands for the fetus. This is being seen in the fetus. Okay, so you have to, by looking at the types of hemoglobin, I think it is becoming clear for you all children that during the phases of growth, there are different sorts of globin chains which are present in the hemoglobin of your RBCs. As you see that in the fetal stage, we do not actually have the A type. A type is going to usually contain two alpha chains and two beta chains, as we have seen earlier. But if you look at these hemoglobin F type, this is going to contain two alpha chains, and instead of the beta, they are going to have two gamma chains. Okay, and even in the adult stage, we have minor fraction of another type of hemoglobin, where you can see that they have two alpha chains along with the two delta chains. Okay, these are the different types of ones. Besides beta, gamma, delta, we even have epsilon also. So we are not considering those, just consider that these are the types of the hemoglobin, and generally this is being found in the adults. This is also found in adults, but in a very minor fraction. Okay, only the minor portion of your blood, adult blood, is going to have HbA2 type. Okay, which is going to have two delta chains instead of the two beta chains. And this is about the normal thing. And when speaking about the thalassemia, I spoke about this is going to be an autosomal recessive disorder. Autosomal recessive disorder. And another important thing for the examination point of view to remember is going to be, this is going to be a quantitative abnormality. Quantitative abnormality of the production of the globin chain in your hemoglobin. Okay, now these, I think I have put up the important things to remember. The first is going to be this, and the second is going to be this regarding the thalassemia. Now, coming on to the details of the thalassemia, children, since I spoke that this is an autosomal recessive disorder, autosomal chromosomes are the vegetative chromosomes, right? So we know that we have 46 number of chromosomes, amongst which the 23 are from the mother and the 23 are from the father. So one few of them are the maternal and few of them are the paternal chromosomes. And amongst this, it might be like 22 are the vegetative chromosomes, with one is going to be a sex chromosome, either X. And if you're going to look at the other one, not this, but if you're going to look at the father, the paternal, it is 22 vegetative or autosomal chromosomes with one of the sex chromosome. And you can see what I have written here is actually a male, right? It is resulting in 44 plus XY. It's going to be a male child. Okay. So remember that we are talking about these vegetative chromosomes. So vegetative chromosomes, you see that you're going to get one from the father, one from the mother, right? For example, if it's going to be the first chromosome, so the first chromosome is going to be one from the mother, one from the father, you're going to get, right? So it is the father, it's the mother. So you can see that it's going to be this pair, right? So we are going to see 46 is nothing but what we are having the two pairs, right? It's going to be 23 and 23. So you can see that the first chromosomes, the second chromosome is from the mother and father. The third chromosome is going to be again from the mother and father. So you're going to see the pair of these autosomal chromosomes which are being present. So now I'm coming into the details of the thalassemia, which is an autosomal recessive disorder, children. So remember that it becomes important for us to understand now that where are these genes present which are responsible for what you call is production of these globin chains of your hemoglobin. Remember that we are going to have the chromosome number 16. Okay, so the chromosome number 16, you're going to get one of it from mother and one of it is going to be from the father. So chromosome number 16 plays a major role because it is comprising of the genes which are responsible for the production of the alpha globins, alpha protein chains of the hemoglobin. So one is coming from the father, one is coming from the mother. Okay, this is a chromosome pair, that is 16. And you can see that you can either call these as alpha 1, alpha 2, and alpha 3, and alpha 4 to make the topic easier now. So remember that the chromosome number 16 is comprising of the two copies of the genes which are responsible for the production of the alpha chain in your hemoglobin. So one chromosome from the father, one chromosome from the mother. So totally how many genes do you have? There are totally 4 genes for the production of the alpha globin chain. Is it correct? Okay, this is going to be the one that you need to remember. Okay, children. In the normal adults, we have spoke that in the normal adults we are going to have 2 alpha chains and 2 beta chains. So how many of the genes are responsible for these 2 alpha chains? Is 4 genes, each from your a pair from your mother as well as a father 16th chromosome. Okay, this is the one. Now coming on to this particular thing is going to be the normal one, which is being called as hemoglobin A type, which is an adult type, right? Now, for instance, if you are going to see that because of a point mutation, because of a point mutation where one single nucleotide will be what we call is going to be replaced by some other nucleotide. Okay, so this will be a point mutation. If at all, just because of a point mutation now, if the one of the gene is being deleted, or just I can call it as just because of a mutation process, if one of the gene which is responsible for the alpha globin chain production, so here I'm drawing only 3 of them, one is being deleted. Okay, so clinically how do we write this is dash alpha alpha alpha. This we write it as alpha 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 from the mother and the father. One of the gene is being deleted here. Only 3 are being left over, right? Only 3. So here you can see that here this person, remember that in the autosomal recessive disorder, in an autosomal recessive disorder, a person has to be carrying both the chromosomes, both the autosomes have to carry a diseased gene. Only then a person is going to be diseased. Okay, let me take yeah. So if they are having, if this is going to be a diseased gene, if both of them carry, only then the individual is going to be diseased. For instance, this is a diseased gene, diseased, okay? If there is something like I told you that if the mutation is going to be only in one, but if it's going to be the 2 of them, I'll show you the conditions. So for instance, if a person with what we call as the autosomal chromosomes like this, and if a person is going to carry one diseased gene, and if he's going to carry one healthy gene, this is a healthy gene, this is a diseased gene, then this person does not show any symptoms. He will be asymptomatic. He will be asymptomatic, and this person will be acting as a carrier. Okay, no clinical symptoms is going to show. It's going to be only when the person is going to have both of the conditions, both the chromosomes, if they are going to contain a diseased gene, or if it's going to be mutated in the both the chromosomes, only then you can see the condition where a person is going to be diseased. If not, he is going to be regarded as a carrier. Okay, children, here you are seeing that only one of the gene is being mutated, and so obviously this person is going to be regarded as carrier, who is going to be asymptomatic. Asymptomatic. Okay. And remember that here there will be no clinical symptoms at all. But if at all, this is going to be the normal individual, right? For instance, if I'm going to speak about another condition where if these are the pair of autosomal chromosomal number 16, and if at all, if the alpha chains because of the mutation, if both the mother as well as the father chromosomes contain only a single gene for the alpha, because normally we require two, right? Two genes from each parent. But here, because of mutation, they lost, and we are having only two genes here responsible for production of the alpha chain. So, what do we call this condition as? This condition is going to be called as a what you call is this is also going to be a person who will be normal, but this is going to be called as a we call it as alpha thalassemia trait. Okay, alpha thalassemia trait we are going to call. Children, this can be either cis or this can be trans. Okay, it can be of cis and trans types. So what is cis and trans? Let me show you. For instance, if these are the chromosomal number 16 from both the maternal and the paternal side of an individual, the two genes which are being mutated here can be either on the two different, or it can be on a single one, right? For example, here, this one is having both the normal genes for coding for the alpha globin chain, but one of the parental chromosome has got mutated and they have lost two genes. Okay, this can be the condition where you can have the two genes. Here also you have two genes. So if you lose on one single chromosome completely, then we are going to call this as a cis condition. If you're going to lose a single gene from both of them, then we are going to call it as a trans. Okay, but both of them are going to be regarded as a alpha thalassemia trait we are going to call. Okay, these people are also normal. Okay, quantitatively you can see that the globin chain production is going to be less here, but they are very normal, and sometimes they may even develop anemic conditions. Okay, they are going to develop the anemic conditions in this regard. Next, you can see another case, children. So what we are trying to do is, due to the mutations, you have seen this condition. Due to the mutations, you have seen a condition where two of the genes are being deleted off. And there is another condition, which is a third condition, where you can see that the chromosomal number 16 here, you can see that the number of genes being mutated will be three of them are going to be mutated, and the one of the parent has the chromosomal number 16 coming from one of the parent is mutated in such a way that they don't have carry any gene, and here only they carry only a single gene. So there are three different there is three genes coding for the globins have been deleted here, and we have lost completely three genes coding for the alpha, right? Only one is present, so only one is a functional gene here, only one functional gene. Okay, and here you can see that there are no enough quantitatively, the globin chain is has got reduced completely. Okay, and remember that here these persons are not going to have enough binding of oxygen, because we need alpha chains and the beta chains togetherly with the heme groups and the iron groups to have a very good binding with the oxygen, right? But here quantitatively we have reduced the alpha chain, due to which what happens is the oxygen binding capacity is going to get reduced. The oxygen binding capacity of your hemoglobin is going to get reduced. Okay, this is very, very important. The binding capacity has got reduced. And children, there will be another sort of what we call is a mutation, where you can see the fourth type of mutation, where you can see that all the four genes are being lost. Four genes of coding for the alpha are being lost completely. This is going to be a very, very severe loss, where you don't have the alpha chains at all. This is going to lead to the death of the fetus if they suffer. Okay, if they are going to have a mutation, and they can't even actually have the what they call is no birth being expected, because the both the parental chromosomes are being mutated in such a way that all the four genes are lost. There is no alpha. And remember, children, I told you that at least you can see that in the fetal stages, if there is no beta, it's okay, because the fetal stage they are going to have the gamma chains. But you have lost the two alpha chains also, which is going to lead to the reduced oxygen, due to which the child, not the child, but the fetus is going to die. This is going to be about the alpha thalassemia, because here the alpha globin chains are being affected, right? So we call this as alpha thalassemia. Alpha thalassemia, this condition. Did you understand? So next one that we are going to see is going to be the beta thalassemia. Beta thalassemia. Remember, children, alpha chains, the coding is being done by the genes present on your chromosome number 16. Whereas if you're going to look at the beta globin chain, the gene is present, a single gene. There we had two genes, right, on a single chromosome. But here, if you're going to look at the chromosome number 11, this is very important now. That was chromosome number 16. This is chromosome number 11, which is going to bear the single gene responsible. So togetherly, there are two genes which are responsible for the production of the beta globin chain, because these are autosomes. We have pair of autosomes, right? So single gene on a single chromosome, but a pair. So we have two genes responsible for this. Remember, if at all, because of a point mutation, this is going to be a point mutation, where a single nucleotide is going to be replaced. So you are going to lose the gene of this particular one, which is responsible for the beta globin. So now let me draw the conditions and show you that there will be a condition because of the point mutation, where one of this is going to be replaced by another. So you have lost this, you have lost the gene from this particular chromosome. And this is going to be called as beta minor thalassemia. Beta minor thalassemia. Because you are having only one gene being deleted, right? One gene is being deleted, but you have another gene which is responsible for the beta globin chain. So this person is also going to be asymptomatic. Asymptomatic, and they also develop anemia. And the way it's being checked out is they perform electrophoresis. Okay, and what they detect is if in the electrophoresis the hemoglobin type is A2, because this is a minor type, and we have already lost the hemoglobin A type. Okay, because hemoglobin A is with the two alphas and two betas. And what is hemoglobin A2, children? What is hemoglobin A2? Hemoglobin A2 is going to be with the two alphas, and two deltas here. Okay, so the major portion of the hemoglobin that is found in the RBCs would be hemoglobin A2, which is coming into the role to play the lost action of the hemoglobin A here, because this is has got a reduced activity because of the production of less quantity of the beta globin chain, right? So who will come into the action now? The HbA2 comes into the action. So by the electrophoresis, what we could detect is the HbA2 has got the major portion of functionality, and it has increased, by which we come to know that, okay, there is something wrong with the HbA. So by electrophoresis technique, they detect that, and this is going to be sometimes mistaken as a anemic conditions because of the iron deficiency also. So this is going to be a one single gene deletion. Okay, children, that is going to be the beta minor. And remember that there is going to be another type also. This is going to be the first type, which is being called as a beta minor thalassemia. There's another one where you can see that both the parental chromosomal number 11, if they're going to get mutated and point mutated, and if they're going to lose both the genes, see, this has got mutated, this has got mutated, and they have lost the healthy gene which is responsible for the production of the beta globin chains, then this condition will be called as beta major thalassemia. And remember, children, in infants, for instance, if you're going to look at infants, in the infants, what was the hemoglobin that was comprising of what are the globin chains? The globin chains were, what was it? It was being called as hemoglobin fetus, right? What were they comprising of? 2 alpha and 2 gamma. So if there is going to be a loss of beta, right? Will there be any major causes or major symptoms in the infants? No, not at all. So what is happening is during the late fetal development, there will be no problem because they are using the gamma chains. But after birth, they are going to become symptomatic. It is going to cause a big problem for the children who have took birth. So now what happens is the child is going to suffer with a very severe anemia, very severe anemia they're going to suffer with, and after the birth they need to go for blood transfusion. So in the infants there is no issue, right? No issue. But after birth, what is going to happen? After the birth, the child is going to suffer with anemia, because as the child takes birth, what happens is slowly the gamma chains are being kept aside and the beta chains are going to be synthesized, and the HbA types are going to develop far better than the HbF. HbF is only seen in the fetus, right? So now what happens is the child suffers with anemia, so he has to be transfused with the blood. So blood transfusion is must for this child now. Okay, and this is also not going to be good because blood transfusion, very regular blood transfusions is going to lead to the iron accumulation in the muscles and the tissues, which will lead to death of the child. So by 15-20 years they're going to lose their life. That is beta major, which is very important. And remember, children, the beta major, what happens is the biggest issue, the symptoms that we are going to see is, since there is less production of RBCs, your bone marrow is going to try to produce many of these RBCs. So what happens is the bone marrow size is going to expand. Okay, and even the facial skeletal as well as the other parts are going to get, especially the facial skeletal appearance is going to be very abnormal because of the bone marrow expansions. Abnormal. This is very important, and they develop the chipmunk faces because of the abnormality. They develop the chipmunk faces also. And remember, since the bone marrow is failing and expanding, your liver and your spleen, these are going to take over the production of RBC. So now they are taking over production of RBCs. So they are going to swell, and that is going to lead to the hepatosplenomegaly, which is swelling of these organs. So that is going to be about the beta thalassemia, where the genes, because of the point mutation, they can lose either one of it or both the genes are going to be lost, and it is going to be a very severe anemia, leading to the abnormalities in the facial skeleton as well as the swelling of your bone marrow, your liver, as well as the spleen. I hope, children, you all have understood. So before I wrap up this particular topic, I will try to show you a few of these slides, what I have, by which you can understand the topic more. So let me show you the chromosomes now. So as I told you that the chromosome 11 is going to contain the beta globin genes here, right? And this is a pair of autosomal chromosomes from both the parents. So you have two genes which are responsible for the beta globin. And next we are going to have the both the chromosomal number 16 from both the parents, each chromosome having two copies of genes here. So totally four genes are responsible for the alpha globin. Togetherly, they make up two alpha, two beta chains in a hemoglobin protein, that is a globin chain. Okay, I hope this is clear for you now. And next, let me come and show you, as I told you, that autosomal recessive disorders are where you can see a person becomes diseased only when both the chromosomes are going to have a diseased defect. Okay, so autosomal recessive disorders where you can see that in the beta thalassemia the point mutations are on the chromosomal 11, but in alpha thalassemia there is no point mutation but there is gene deletion. This is what I wanted you to clear off in your brains, that point mutations are seen in the beta thalassemia, and in the alpha thalassemia it's going to be gene deletions. And remember, children, if both the parents are going to carry, that is, both the chromosomes combination in the child is going to be having the defect, then only the child is going to be affected. Okay. For instance, if you can see here, the portion, the trait, the diseased trait, because both the parents are having here. Uh, both the parents that you're seeing on your screen are the carriers, but they have donated the half of the disease gene. Means togetherly the both the chromosomes are having the defect, so the 25% of the children are going to develop the thalassemia. If it's going to be one of the healthy from the parent and one of the chromosome is having a defective gene, then we are going to have a 50% rate. Okay, and so they are also regarded as a carrier. But if it is going to be both the healthier genes from the parents, then the child is going to be unaffected, a normal child. So 25% is disease, 25% is normal, and 50% are going to be the carriers in this particular diseased autosomal recessive disorder. Okay, children, I hope I made this point very clear for you all, children. I hope you all have understood this particular autosomal recessive disorder, which is a quantitative abnormality seen in the production of the globin chains of the normal hemoglobin. And I hope you also understood the hemoglobin part structure and how the proteins are being embedded with the heme as well as the iron parts. If you like this video, do please hit the like button. Do please share, subscribe, and try to comment in the comment section how you liked and how this video has helped you in clearing the concept of the autosomal recessive disorder. Thank you all. Thank you.